filling in, but it is 1:01. So I'll start by introducing our speaker for today, Dr. Kevin Myers, speaking to us from Bucknell University. And so Dr. Kevin Myers got his PhD in psychology from Duke University. He then went on to do a postdoc at Brooklyn College with Tony Sclafani, where he studied carbohydrate and fat appetite. He started his lab at Bucknell University in 2001, where he's now a full professor. And since he opened his lab, he's really consistently published on the biopsychology of food intake. And it's really notable that he's a fantastic mentor as well as scientist. And if you look at most of the papers that came out of his lab, he has co-author undergraduates on almost all of them. And his mentorship and training of these undergraduates has been so fantastic that we've actively recruited now multiple trainees from his lab to join our feeding group in the Philadelphia area. Outside of his lab, Kevin's very active in the feeding community, especially through his participation in the Society for the Study of Ingestive Behavior, which is where I first met Kevin over 10 years ago now. He's currently the acting secretary for SIB, and more informally, he is the SIB member most likely to be out socializing until 5 a.m. the night before giving a talk. And so, Kevin, I'm sorry that we weren't able to take you out for a beer last night to continue that tradition, but we're so glad that you're able to join us virtually to tell us more about flavor nutrient learning in the obesogenic environment.
Thank you, Amber. And thank you, everybody, for turning out like this. I mean, I'm scrolling through a couple pages of Zoom here and just seeing this who's who of some of my own heroes and role models. So I'm just really grateful that you turned out like this. And I'm grateful and appreciative to Amber and to Monell for hosting me. I'm going to start by saying what a treat it is for me to present at Monell. I have been a fan of Monell Chemical Senses Center literally since I was a teenager. A little Kevin Myers trivia for you. Some of you know I am congenitally anosmic. And so I've been fascinated by the chemical senses since I was a little kid. And I also grew up in the suburbs of Philly in Delaware County, right outside of West Philly. And so as a teenager, heading into Center City with my friends to hang out, I remember one day noticing at 3500 Market Street, this big golden nose, the Monell Chemical Senses Center. There's a whole building full of people who study taste and smell. And so I had the good fortune of, in 1994, spending a short time, just a little less than a year, working as a technician at Monell for Beverly Cowart. And that's when I met people like Beverly and Julie Manella and Gary Beecham, Paul Breslin, Marcy Pelchat, people who really became role models for me. And I'm so appreciative of that. I kind of think of this as my nature paper, right? So in 1994, Paul Breslin published on a unitary sweet taste quality for the basic carbohydrates. And I don't know if that's my data, but I was a participant in that study. And then some 20-some years later, my friend Lindsay Shearer and I published recently on rats' ability to discriminate oral sensory qualities of glucose and fructose. And it was seeing Paul Breslin present on that in 1994 that actually planted a curiosity about that in the back of my mind. But anyway, it's a thrill to be here. So Ms. Donut. Talks like this about appetite and overeating commonly start with a picture of something appetizing and delicious, like why are we compelled to overeat these high-fat, high-sugar stimuli? But you start with something a little more challenging. Those of you in Philadelphia may recognize this as Scrapple. Those of you from outside Pennsylvania who are calling in, let me tell you a little bit about Scrapple. It is a breakfast meat loaf made of the leftover scraps when you're done butchering a hog. And this is a curious substance to me because I have never in my life seen an adult taste Scrapple for the first time and like it. Yet to those of us who grew up with it, This is, I find scrapple delicious. And I gave up eating meat a while ago, but still looking at this photo, my mouth is watering. I'm craving it. And if we were to go to the opposite side of the globe in Southeast Asia, you would find durian fruit, which the flavor of durian fruit, if you've never tried it, is best described as a combination of melon and custard and rotten onions and dirty sweat socks and burning garbage. People who grow up with durian come to love it, but to outsiders, so you see it, there's a durian food festival. You can get durian milkshakes at McDonald's in Thailand, but to people before, the flavor of durian is so repulsive that in the Singapore metro system, you're prohibited to smoke or carry explosives or durian fruit. And hotels in Thailand will actually fine you for cutting open a durian in the room because outsiders find the flavor of durian to be so repulsive. And so a question I've long been interested in is, how is it that a wide variety of human cuisines have foods that people who grew up with that food come to love it, but to outsiders, those foods may seem at best neutral, at worst actively repulsive? And so the name of the game here is experience-dependent plasticity. Humans are generalist omnivores, and humans live virtually everywhere on the globe. And the rats and mice that we use in the lab to model our behavior live everywhere that humans do. And the reason they're capable of doing that is because of our evolutionary history as generalist omnivores. We're capable of making a diet from whatever happens to be available in the environment that we're born in. And because of that, there's no way that our genome could anticipate what's going to happen to be available where you're born. If it's going to be, shrimp and cranberries, or if it's going to be buffalo and elderberries, or if it's going to be who knows what you would find in the Sonoran Desert. But the work of people like Julie Manella has shown for years that we come prepared with only a few basic hardwired taste preferences and avoidances, but most of our likes and attraction and interest in foods and flavors is based on a history of learning through experience. So what I'm going to talk about today is one specific type of experience-dependent plasticity, one specific way that our attraction to particular flavors is shaped by experience, and that's flavor-nutrient learning. The way that we come to learn to associate initially arbitrary flavors of foods with post-ingestive nutrient sensing and the type of condition shifts that produces in our response to those flavors. And I'm also going to then talk some about how this learning system, which evolved to enable us to capitalize on unpredictable food sources, now how that might be intersecting with the modern obesogenic environment to cause or to contribute to overeating and diet-induced obesity. So quick primer on Pavlovian conditioning of responses to flavors. That everybody's probably familiar with the idea of you consume a food with a flavor and then get sick afterwards, and you learn a Pavlovian association between the food and the illness and form a strong disgust response to that flavor based on that learning. Well, this type of learning works in the opposite direction as well to produce preferences for flavors. So, the first time you ever ate an apple, as a simple everyday example here, there's nothing inherently attractive to humans about the flavor or aroma of apple flavor, but you eat an apple and it's accompanied by the sweetness of the sugar that's in the apple. And that produces a type of associative learning called flavor learning, that the flavor of the apple itself is a conditioned stimulus, that its value is enhanced by its learned association with the inherent attraction to sweet taste. But eating a food is also accompanied by post-ingestive nutrient sensing. And so the type of learning that I'm going to focus on is flavor nutrient learning, which is the way that the flavor becomes to be associated with the post-ingestive nutrient effects of the food to produce changes in how we respond to that flavor, an increased attraction to it, and potentially an increased palatability of it. So in a lab rodent model, we need to be able to separate those two types of learning. So how do we study the ability of rats to, say, associate an arbitrary flavor with post-ingestive nutrient sensing of, say, for instance, carbohydrates, unconfounded by the positive taste properties of that carbohydrate? Well, we do that. Was a system that was developed by Tony Sclafani, and Karen Ackroff helped Tony with a lot of this work. And now I've duplicated this system in my lab, the electronic esophagus system, where we can give the rat a flavored solution to drink, for instance, a Kool-Aid flavored saccharin, that itself is not providing any post-ingestive nutrient value. And the electronic lick detector is monitored by computer, which is controlling an infusion pump, which is delivering through an intragastric catheter, either water or a fat solution or carbohydrate solution. And now that allows us to independently manipulate the taste and the solution with the post-ingestive nutrient that is sensed in the gut. And so this is a picture of what this picture is from Tony's lab, and literally this box is now in my lab. Fantastic donation that I received from Tony at the time of his retirement. But here you see the rat drinking an arbitrary flavor, for instance, cherry Kool-Aid, that the rat would have no innate instinctive attraction to, accompanied by an intragastric infusion of sugar. And so we're looking for the rat's ability to learn to associate this flavor stimulus experienced orally with the post-ingestive nutrient sensing that's happening in the gut or somewhere post-absorptively. And so what a typical training paradigm would look like is alternating sessions between two distinct flavors that the rat can clearly tell the difference between these flavors, but initially would have no strong attraction to one over the other. And so you would counterbalance this across rats, but say, for instance, an individual rat would get trained with, say, cherry Kool-Aid, and we're going to call that the CS plus flavor because it's accompanied by the unconditioned stimulus of, say, a post-ingestive infusion of glucose, alternating with sessions of a CS minus flavor, a conditioned stimulus flavor that is unpaired with post-ingestive nutrient sensing. It's accompanied just by a water infusion. And so there are a couple of pretty striking behavioral changes that you start to see pretty quickly with this conditioning. One is a condition shift in acceptance of the nutrient paired flavor. So over repeated training sessions, you start to get larger and larger and larger meals of the flavor that's being accompanied by post-ingestive glucose sensing in this case, which is a little unusual, thinking that, well, the post-ingestive nutrient action should be causing satiety. And so why are the meals getting bigger and bigger if you're learning that this is accompanied by a satiating infusion? Well, you're producing a conditioned enhancement of flavor acceptance. And then in a 2-bottle choice test with no nutrient being delivered, so behavior here is based entirely on your learning and memory about prior experiences with this flavor, you see a very powerful preference for the flavor that was previously paired with nutrition. And so a couple basic behavioral phenomena that we see is that flavor nutrient learning to associate an initially arbitrary flavor with post-ingestive nutrient sensing, alters our preference for which foods we are attracted to, and alters acceptance, that is, increases in how much of that food you're interested in consuming. And this is a very rapid form of learning. For years, animal learning specialists thought that conditioned taste aversion was unique as an example of Pavlovian conditioning because it could be learned so quickly in a single trial. Well, flavor nutrient learning can be learned in a single trial as well. So these are rats that are getting just a single 10-minute a day session of drinking unflavored saccharin, so just a little bit of sweetness. And it's being accompanied by an intragastric infusion of water. So there are no calories involved here. And then on one day, they get a 10-minute session with either coffee or vinegar flavored saccharin, accompanied by a glucose infusion. And on another day, the opposite, vinegar or coffee paired with water. And then after that single 10-minute meal of either coffee or vinegar flavored solution paired with an intragastric and glucose infusion, a couple days later, you see in a 2-bottle choice test, a very strong preference for the flavor that was one time paired with glucose over the flavor that was paired only with water. And that preference persists even though the glucose infusion is no longer being delivered in these repeated training sessions. So this is a pretty robust type of learning. And so this is some new unpublished data, and I want to give a shout out to my former student, Jamie Carty, who was a Bucknell undergraduate, who's now down there at Penn as a technician in Nick Bentley's lab. We found that this type of learning is relatively insensitive to homeostatic negative feedback signals indicating energy surplus. So in this experiment, what we did is we trained rats in this standard flavor nutrient paradigm, either under conditions where they were getting a lateral ventricle infusion of saline into the brain, or a lateral ventricle infusion of leptin to mimic elevated leptin levels that would result from increased body weight. And then later on, with no drug or no hormone on board, gave a 2-bottle preference test for the CS plus and CS minus flavors. And we found that leptin levels, elevated leptin levels in the brain at the time of training did not interfere with the strength of this conditioning. And if you take this flavor here that they previously learned, this flavor nutrient preference for this glucose paired flavor with no leptin on board, now retest them in this preference test, again with either ICV saline or a treatment with ICV leptin. Adding leptin in the brain at the time of training is not diminishing the behavioral expression of their preference for a flavor that signals nutrient consequences. So we're finding that this, and this is consistent with my work and Tony Sclafani's work, showing that the level of food restriction, the rats are relatively insensitive to having recently eaten doesn't necessarily diminish rats' preference for a nutrient paired flavor. And so it leads me to wonder about the degree to which this flavor nutrient learning is an obesogenic influence, given that we're in an environment where we have a lot of opportunity to consume high calorie foods. And this is a type of learning that is going to steer preference towards flavors that predict high calories and also increase meal size of foods that we learn are high calories. Gotten interested in studying the role that flavor nutrient learning might play in the modern obesogenic environment. Okay, so first I'm gonna try to talk vaguely physiologically about something that I really think of as a psychological issue. A lot of different groups have done work that I think is cool and fascinating about trying to identify when nutrients are sensed in the gut or at some post-absorptive step after nutrient ingestion, what are those reward signals that are fed back to the brain to form this associative learning or this preference for the flavor that predicted them? So, you know, there's this long cascade of physiological events that happen on this time scale during and after a meal, and these feedback signals from the gut and from metabolism can travel to the brain through several different routes. So Tony Sclafani in particular, but other groups too, have worked on, say, trying to knock out specific gut feedback signals to see the degree to which it interferes with flavor nutrient learning. And I've taken a different route as a psychologist. I've tried to ask rats about their feelings. Getting them to lay still on the little couches to tell me about their feelings is the hardest part of the experiment. But I've tried to take a psychological approach. Instead of physiological manipulations, can I get rats to tell me in their behavior at what point in time are they noticing the reward feedback signal? And that would give us a clue as to its physiological indicator. So asking rats when, during or after a meal, are they noticing this physiological feedback? And how to do this relies on a basic principle of temporal contiguity in Pavlovian conditioning. So say that you're training rats in some arbitrary Pavlovian conditioning and you have two different conditioned stimuli, like a light and a tone, and they're followed by an unconditioned. Well, the principle of temporal contiguity in Pavlovian conditioning is the unconditioned stimulus is going to become most closely associated with the CS that immediately preceded it in time. And so if you were to do this training with the unconditioned stimulus delivered here, it's now going to become associated with CS1 and not CS2, which now represents backwards conditioning. Well, could I do this type of thing in the flavor nutrient learning paradigm? What we did is we trained rats with meals always had two flavors, the early flavor and the late flavor, both of which are being accompanied by infusion of glucose. And asking then, this could tell us something about when in time the rats are actually experiencing the relevant post-ingestive signal that's producing this reward, based on which flavor do they learn a stronger preference for. So if the relevant reward signal is occurring after some long delay, they should learn a stronger preference for the late flavor, not the early flavor. But if the relevant reward signal is happening relatively early in the meal, perhaps then they would learn a stronger preference for the early flavor, not the late flavor. And so this is the training paradigm where this looks, I think, more complicated than it really is. Rats are alternating between two different types of sessions. In positive sessions, the meal is accompanied by gut glucose infusion. And in negative sessions, it's just accompanied by gut water infusion. So you have two different flavors that are accompanied by glucose, but one of them is occurring early in the meal and the other later. And we're going to then see, relative to these control flavors, which flavor does the rat learn a stronger preference for? Well, it wouldn't be science if it didn't turn out a completely haywire way that was a little bit different than what all of us expected could happen. Turns out rats learn a preference for both of these flavors. That they learn a strong preference for the flavor occurring early in the meal and a strong preference occurring for a flavor late in the meal. But this isn't a wash. I think that this provides us some interesting insight to an idea that there are multiple post-ingestive signals happening on different timescales. And the evidence for that is that what they're learning about these two different flavors, they're learning something different about the early flavor and the late flavor. Because when you test them food deprived, they show a strong preference for either flavor. But when you test them recently satiated, they now show only a preference for the flavor occurring late in the meal. So that difference in the behavioral expression of the preference suggests to me that they've learned something qualitatively different about these two flavors. And I would note that this is a phenomenon that many of us see and experience in our everyday lives. Our cuisine typically has foods that occur early in a meal, like entree foods, and foods that occur late in the meal, dessert foods. And the type of learning that occurs is you acquire preferences that are hunger dependent. So now when hungry, you're interested in seeking out meal foods, but you also acquire preferences that are hunger independent. If recently satiated, you're no longer as interested in meal foods, but now interested in the flavors that you typically find in dessert foods. Another aspect of this that I think is interesting or a bit provocative, and I think, Amber, hopefully relevant to your interests, is you see a different pattern if the nutrient that you're infusing is fat rather than glucose. And so there's a variety of different evidence that the gut feedback signals that could produce reward and flavor nutrient learning in the case of fat are qualitatively different than those produced by simple carbohydrates, that they are communicated through a different route and perhaps on a different time scale. So when we do this same training where there's a flavor or a meal with two flavors, both accompanied by fat, rats no longer learn a preference for the flavor occurring early in that meal. So when tested either hungry or satiated, they now show a preference only for the flavor that was occurring at the end of the meal. And you see the same thing when they're tested satiated. So what this is leading me to speculate is that in the case of arrival of nutrients to the gut, that's generating multiple distinct feedback signals that are happening on different timescales, and that they can all support flavor nutrient learning, but the type of learning that they produce may be qualitatively different. So in this case, the hunger independent learning instead of just the hunger dependent learning. And so I think that this work helps to challenge a long-standing dogma that's existed in the field of appetite biopsychology for a long time. The idea that the things happening in the mouth are primarily positive feed-forward signals, and that things happening in the gut are primarily negative feedback signals. That, sugar in the mouth promotes intake, and sugar in the gut serves to suppress and bring meals to an end. This term coined by Tony Sclafani, appetition, in contrast to the better known processes of satiation, refer to these positive feed-forward signals that come from gut nutrient sensing. And my behavioral work is indicating, at least, that there are multiple appetition signals occurring on a couple different timescales. There are rapid within meal signals that lead to learning of hunger dependent preferences, and then more delayed feedback signals that can also support preference. But this would help us understand why many experiments done over the years trying to knock out single gut feedback signals like vagal signaling or CCK or GLP-1, that trying to knock out single signals has often shown minimal effects at all on flavor nutrient learning. I think a reasonable explanation would be is because there are multiple somewhat redundant gut feedback signals. Okay. So become interested in this rapid onset signal. And you can actually see behaviorally, in the very first time that rats are drinking a flavored saccharin solution and getting a glucose solution to the gut, you can see within minutes their behavioral response to it. So here we have rats being trained over days with just different flavors of saccharin sweetened Kool-Aid accompanied only by water infusion. And eventually what you end up with is this very stable baseline of cumulative licking during a 30-minute session. So this is the 15 minutes when the water infusion is being delivered to the gut, and this is their cumulative rate of licking for saccharin. But then the very first time you give them a novel flavor, accompanied now by glucose infusion, within a few minutes into the meal, you see this positive feed-forward appetition effect. And so this data of mine replicates something that's now been shown by Tony a number of times in both rats and mice, that the cumulative licking for just unflavored saccharin solution or a flavor of saccharin accompanied only by water to the gut. And the very first time, in this case, mice are receiving glucose to the gut, you see an acceleration of licking within a few minutes. And then the very next time, the mice encounter that same flavor that they got in test 1 here, accompanied by glucose. From the first minute, their licking is now accelerated because they've learned, based on that associative experience, of that novel flavor being accompanied by gut glucose sensing. So I want to now look at the link that that rapid feedback signal might play in overeating and its response to diet-induced obesity. And I got interested in that because we know a lot about how diet-induced obesity changes the brain and changes the gut. You get, powerful shifts in reward processes that are going to mediate Pavlovian responses to food predictive cues. And you also get changes in gut morphology that could alter mechanisms of nutrient sensing and communication of peripheral events to the brain. So what I thought was a simple question turns out not to be a simple question, but I think I have some interesting effects, is what effects would you get on this basic flavor nutrient learning in rats who have been made obese on a high-fat, high-sugar diet? And so we used Suzanne LaFleur's model of the high-fat, high-sugar choice diet, where in addition to plain chow in the control group, the high-fat, high-sugar group gets ad lib access to a 30% sucrose solution and lard. And what you see, like on a lot of obesogenic diets, you see rats break out into 2 subgroups. Those that are more obesity resistant and don't gain much more weight than the control group, and those that are much more obesity prone and gain a lot more weight. And then what we're going to do, and I want to acknowledge the work of Hallie Wald, who was, this was her undergraduate senior project in my lab. Hallie's now a PhD candidate down there at Penn in Harvey Grill's group. Again, what we're going to do is just familiarize rats with drinking the flavor of saccharin and accompanied by water infusion to the gut. And comparing these 3 different groups, chow only, or high fat, high sugar, who are more resistant to obesity, or those who have become more obese, in what happens the first time the flavor that's accompanied by delivery of glucose to the gut. And what you see is we intentionally chose an infusion of glucose. This is a relatively low concentration that we wanted to produce a relatively weak feedback effect. So we wouldn't get a ceiling effect. We could see a movement in either direction. And so it turned out to not be a very effective gut reward signal for the control group. But the more obese rats are showing an enhanced sensitivity to the immediate arrival of glucose in the gut. Eventually, however, they all acquire acceleration of licking. So by the 3rd pairing of this flavor with glucose, all of the rats have now elevated their licking. But in a 2-bottle choice test, between the flavor that had been nutrient paired and the flavor that was always paired with only water, they all learned a significant preference for the nutrient paired flavor, but it was actually the most obese rats who are expressing the strongest preference for the nutrient paired flavor. And so this got me thinking about, well, in all sort of ways where you see obesity correlate with behavioral differences, there are complex questions of causality, that this because these are the rats who are eating the most sugar and fat and experiencing the most physiological impact of the onset of obesity. Those impacts could be causing a shift in their responses to gut nutrients. But it could have been a pre-existing difference that, we can't randomly assign rats to be obesity resistant or obesity prone. God makes them that way. And so could it be a pre-existing difference that contributes to the onset of diet-induced obesity proneness? And so what I wanted to ask then is, what if we do the experiment in the opposite direction and just measure rats' individual differences in their susceptibility to flavor nutrient learning and then put them on an obesogenic diet? Would sensitivity to flavor nutrient learning predict subsequent behavior on the obesogenic diet? Okay, well, it's difficult to get individual differences in flavor nutrient preference learning because it is such a strong and robust type of learning that there are almost no individual differences. So this is actually after just a single pairing of flavor with post-ingestive glucose, strong preference for that flavor, and it basically breaks out as these are individual rats. A vast majority of the rats are showing a ceiling effect, and a small minority of the rats are just not quite getting it. But you get much better individual differences in the acceptance effect. So this is the tendency of rats after learning to associate a flavor with the post-ingestive effects of glucose, their tendency to eat larger and larger meals of that flavor. Well, there you get beautiful in a range of individual differences here. So this is the group averages of increased meal size for the nutrient paired flavor, but you see a whole range of individual differences. So what we did is then put those rats on an obesogenic high-fat, high-sugar diet to see, would these individual differences in sensitivity to nutrient-conditioned acceptance correlate with the onset of diet-induced obesity? And I have not published these data yet because I genuinely have no idea what to do with them next. It did strongly correlate in the absolute opposite direction as I predicted. We put rats on the high-fat, high-sugar cafeteria diet, and this is individual differences in weight gain. So you see, on average, all the rats are gaining weight, but say, here's a rat who's gaining not much weight, whereas this is a rat here who's gaining a lot more weight. And so you could look at either the slope of that weight gain or the percentage of weight gain, and this is your range of individual differences. So let's correlate this individual difference in weight gain with the previous individual difference in flavor nutrient acceptance, and this is what you find. That after controlling for baseline liking just for sweet taste in the absence of flavor nutrient learning, you see a relatively strong correlation in the residuals that increased acceptance for increased meal size of a flavor that predicts nutrient is predicting the lowest weight gain on a cafeteria diet. And this makes me think about metabolic conditioning rather than things like palatability and wanting, because the individual differences in flavor nutrient acceptance learning predicted weight gain as much and as well as actual caloric intake on the cafeteria diet. And so, that rats, different rats given the same amount of calories can gain different amounts of weight because of individual differences in metabolic efficiency. Well, that's an effect that we're seeing here. And so it makes me think that individual differences in flavor nutrient acceptance conditioning are not just about increased liking or wanting being conditioned for the flavor. It's something about conditioning of metabolic preparatory responses. And so here I'm taking a cue from work like in Dana Small's group, where she does brain imaging of people after flavor and nutrient learning training. And what she finds is that the strongest brain signal in the fMRI after flavor nutrient learning isn't related to a self-reported liking response. It's most strongly correlated with a metabolic response, that the glucose excursion that occurred during the flavor nutrient training ends up being strongly related to how much of an fMRI signal you get in response to that flavor at a subsequent testing session. So thinking about metabolic preparatory conditioning. Okay, with not a ton of time left, I just want to go now in a different direction and return to the obesogenic environment. These learning abilities presumably evolved in an environment of scarcity that would enable us to make the best use of relatively poor quality food sources that are only occasionally available. And now we have these learning abilities operating in this environment of excess. And a puzzling thing about flavor nutrient learning is, as robust and reliable as these effects are in lab rodent models, it's actually hard to find experimental de novo conditioning of flavor nutrient learning in humans. I saw Martin Yeomans is on the call, and he's has a couple good demonstrations of this, but Martin has also written about reasons that it's difficult in an experiment to do flavor nutrient learning in humans to get good behavioral evidence of changes in responses to flavors that are accompanied by post-ingestive nutrient signaling. And this idea was suggested by my friend and sometimes collaborator, Jeff Brunstrom, about a flavor confusion effect. Because we live in this modern environment surrounded by flavors of foods in processed foods, we have a confusion about what do flavors really mean in our individual learning experience. An example, there was for a long time in our evolutionary history, if you had the flavor of raspberries in your face, you were about to get the nutrient content of raspberries in your gut. And if you had the flavor of almonds in your face, you were about to get the nutrient effects of almonds in your gut. Well, now you can buy raspberry flavored almonds. And so there's potential for confusion here about what does that raspberry flavor actually signal. If you get bored with the raspberry flavored almonds, you can get raspberry flavored granola or raspberry flavored M&Ms. You could also get honey something granola or cinnamon granola. And so we have all these examples of foods with identical nutritional content that have totally different flavors, and foods with very similar flavors that have totally different nutritional content. And so, a good example of this is in goldfish crackers, that the flavor properties of processed foods no longer reliably signals their post-ingestive nutrient effects. And so I took this up the street from my house in my neighborhood. You can buy a coffee now that tastes like your favorite ice cream. So Jeff Brunstrom's suggestion was, does our history of humans with this confusing flavor nutrient relationships in our modern diet impair our ability for flavor nutrient learning? So we tried to model this in rats. Let's give rats this modern diet versus a natural diet where presumably the flavor nutrient relationships were much more intact, and then look to see if this history of ostensible flavor confusion would impair flavor nutrient learning. This was a fun study to run, but everybody in the lab that semester gained a few pounds because this required keeping an excess supply of junk food on hand all the time. So over a period of 3 months, the rats in the flavor confusing processed cafeteria diet we're getting all these processed foods. And we tried to pick foods where there would actually be the potential for flavor confusion. So for instance, several different apple cinnamon flavored foods and several different cheddar cheese flavored foods. Whereas other rats got an entirely natural, minimally processed diet that would preserve the natural, quote unquote, relationship between flavor and post-ingestive consequences. This was a fun study to run. The rats seemed to really enjoy it. And we tried to not make weight gain a factor. So the processed food group did gain a little more weight, but we didn't want this to be an obesity study. We just wanted it to be about experience with different flavors. And what we found is the exact opposite of what would be predicted by this flavor confusion hypothesis. The rats fed the processed modern diet that would ostensibly produce flavor confusion showed the strongest effect in conditioned acceptance now when we train them of a novel flavor paired with gut glucose sensing. And they actually showed the strongest conditioned preference for that flavor paired with gut glucose sensing. And so unlike our initial thought of a flavor confusion effect, what we think we're finding here is actually a flavor expertise effect, that rats with this confusing history have perhaps become better at making subtle distinctions about, okay, apple flavored Cheerios are different from apple flavored granola, that kind of thing. Okay, so just to wrap up then, I see these types of learning abilities as something that evolved in our ancestral environment to enable us to cope with relatively low quality, unpredictable food sources, but that are now operating in this modern environment of excess. And what we've found so far that I can be confident about is complex links between these learning mechanisms and diet-induced obesity, and a skepticism about some existing ideas about why it's hard to find flavor nutrient conditioning in humans. And I'll just leave it that physiological mechanisms remain to be explored, Amber. And so I just want to end by acknowledging with appreciation how much fun it has been to work with all the students who contributed to the work that you saw presented today. And just a tip to any postdocs or grad students on the call that you can have a rewarding career working in an undergraduate environment. And so I'll just also say thank you to you for your attention. And that's what I have for you today. Thanks.
Kevin, thank you so much for a really interesting talk. We're open to questions now. You can use the chat to submit your questions, or feel free to hop on and ask your question by unmuting. But I think I will take the opportunity to ask you the first question.
I'm going to remove the screen share just so I can see more people. Oh, I almost accidentally left the meeting. That would be not helpful. Okay.
All right, we still got you? Awesome. Yeah, so I was really interested to see the differences in the early versus late flavor preferences. And, you know, I'm also interested in this because you saw nutrient differences as well. So I was wondering if you could speculate a little bit about the mechanisms mediating these different early phase versus late phase. Do you think this is differences in gut-brain pathways, or differences in central circuits and pathways, or both?
Yeah, everything I could say about that would have to be entirely speculative. I'd be strongly tempted to turn the question over to Tony because he's done the most work on this. A challenge here is there are unfortunately apparently pretty strong species differences in mice versus rats in sort of the effectiveness of gut feedback signals. And so as a lot of the physiological work, discrete signals has shifted to mice, it makes it a bit harder to make that translation. But, you know, as you well know, because you cited it in your recent talk, work that Tony did a couple of years back on vagal deafferentation and what that doesn't do to impair nutrient feedback that produces preference. And so that's led to investigation of sort of intraluminal sensing of different nutrients, because where into the intestines you deliver the nutrient has a big impact. But there's been a variety of behavioral evidence that in general, at least in rats, fat delivered to the gut produces weaker conditioning, and it takes more experience to produce that conditioning. So say several training trials instead of just a single trial. And so it would be out of my depth to speculate about specific physiological signals. But what I'm interested in as far as pursuing possibilities is the idea that there are multiple signals. That Tony's best evidence, I think, to my reading, about that rapid early onset signal that's happening within the first several minutes of the meal has been linked to the S-GLUT1 transceptor in the intestinal lumen. But then subsequent signals farther down the road, whether they have something to do post-absorptively about the nutrient itself being detected or some metabolite in the brain, it's a question that makes me think of sort of behavioral investigations, but it's I wouldn't have the relevant expertise to talk about specific physiological signals.
Yeah, I think it's a really interesting question. We have a question from Mark Friedman. Mark, if you'd like to unmute and come on screen.
Hi.
Hi, Mark. Good to see you.
You too. I got a sort of a specific question and a general one. One is, and maybe this has already been done, but when you talk about fats, there are different kinds of fats, and most studies, I suspect, use long chain fats, which are, in fact, absorbed fairly slowly through the lymphatic system. Medium chain fats, and maybe Tony's done this or other people, are absorbed very rapidly. I don't know if it's as rapid as glucose, but it might be interesting to see in your timing studies whether medium chain fats actually had an effect earlier. Maybe you have to change your criteria of what early and late is, but that may be something to consider. I don't know, has anybody done medium chain fats?
Okay, so that's a really clever suggestion, Mark. I appreciate it. And yeah, in that way, fat is quite a bit more complicated than monosaccharides and disaccharides. In my studies for fat, we almost always use dairy fat. So we mix basically heavy cream and dilute it because it's pre-homogenized and therefore very inexpensive relative to something like intralipid.
Right.
So I know that Tony, in his studies with fat, have typically used intralipid as a fat source. But the manipulation of different fatty acid profiles, I'm genuinely not sure if that's been investigated.
No, it's a matter of the medium chain fats are absorbed very quickly right into the portal vein. And dairy fats would have some short chain fats, which would do the same, I suspect, as well. So
and that would be revealing, because it would tell us something about, okay, this more delayed effective fat. Is it because the actual signal is something that doesn't start onsetting until later? Or is it just, there is one common pathway that all of these nutrients are hitting, and it just takes fat longer to get there because of its slower absorption?
Right. My bigger question is, what is the evidence that humans overeat because food's more palatable?
Wow.
The direct evidence. I mean, there's a lot of hand waving about it. And it makes sense to everybody, but is there any direct evidence for that?
I'm looking at Martin now and trying to see which direction he's shaking his head. But the source I cite, I think, is Martin's about the degree to which experimental manipulation of palatability produces predictable changes in meal size. But those are all relatively short-term studies.
Exactly.
Yeah, so your point is well taken that this is something that we take to be true, often in the absence of much sort of controlled evidence of that.
Short term, absolutely. You'll eat more food that tastes good than food that tastes worse. Mm-hmm. But long term, over a nutritionally significant interval, I have not come across any direct data that palatability drives obesity, basically. Okay, I'm just checking in for somebody who would be more up to date in the literature.
Well, I think Jeff Brunstrom talks about analysis of large-scale data sets, where if you just ask, and unfortunately, I'm giving you this secondhand based on hearing Jeff talk about it, so I can't give you a citation. But large-scale data sets, where if you look at responses from hundreds, thousands of people, just ask people their favorite foods and food frequency questionnaires. And there is a relatively strong correlation in food frequency and liking between energy density and food frequency. But yeah.
But the issue is whether it's palatability or metabolic effects that are important, then that doesn't answer it.
Yeah, and my thinking about flavor nutrient learning has shifted steadily over the years in initially thinking about what flavor nutrient learning does. It makes flavors more palatable. And in my work with Tony, when I was a postdoc, what we showed was that under certain circumstances, a nutrient paired flavor becomes more palatable, but not always. We were able to do training with a couple different paradigms where rats would show very strong increased acceptance and preference for a flavor, but no taste reactivity evidence that they were treating the flavor as more palatable. So that led me to think about, well, it's more about incentive salience and sort of attraction to flavors, that nutrient paired flavors aren't more palatable, they're more interesting. But then I think this whole new unexplored area is about metabolic preparatory conditioning.
Okay, thanks.
Awesome. We have a question from Anne Marie and then Martin.
Hello. So
hello.
I wanted to talk about the early and late pairings again. So in the second flavor, the flavor that got the later pairing, it sort of got the experience of an early pairing because it was getting glucose in that moment and the sort of later pairing from the previous infusion. So it got kind of a double whammy of learning effects there. And when you talked about that, you talked about it as like the cupcake after dinner, that when you're not hungry, you might be attracted to pasta, but when you are full, maybe you're going for this late flavor pairing. But did you ever ask the rat that? Did you ever pair the early paired flavor versus the late paired flavor?
Yes. Yeah, I cut that slide from the talk so I wouldn't have to segue into this question as kind of a speed bump right in the middle of the talk. It's complicated. So rats do not prefer the early paired flavor over the late paired flavor. They prefer both flavors, but early and late glucose paired over their corresponding CS minuses. They don't show a strong preference between the two, except for in the fat study. In the fat study, they show a strong preference for the later flavor over the early flavor. Is that your question?
Yeah. In the fat study, they really only developed a preference for the later flavor, right?
Right, yeah.
And develop one. So it's not that the later flavor in the glucose study, there was a stronger preference. It's just that that preference withstood feeding.
Correct, yes. And what was frustrating about that is that it wasn't then hunger dependent in that pairwise choice, which kind of threw a wrench in the works. So I will openly admit that now, but I didn't want to lose your confidence at that point earlier in the talk. There is something, since you're bringing it up, that I want to say about this. So going back to the screen share for that experiment, the reason that this delay is 4 minutes here, in an initial pilot experiment, that delay was only 30 seconds. We paused the session, the bottles retract, made it just long enough for enabling us to switch the bottles. And what would happen in that case is that when the bottles would reinsert, the rats would reject this late flavor. That it was as if they were acting as if by the end of this 8th minute here, that they're already attributing the glucose that's arriving in the gut to this early flavor, that when you take it away and replace it with a new flavor, they reject this new flavor. But they don't do that when there's only water showing up in the gut. You take the early flavor away and replace it with the late flavor, and they just keep on drinking oblivious. I don't have the slides here, but I did publish subsequently an experiment showing that that effect, essentially. If there is glucose showing up early on, rats decrease their intake of the later flavor as if they are attributing the glucose that's arriving to the early flavor, but they're not bothered by a flavor switch when only water is showing up in the gut. It's kind of I think of it as a sort of old Stanley Schachter misattribution of emotions experiment.
Martin?
Hi, Kevin. Really nice to hear you speak. And it's fun to join in from the other side here.
It's great to have you.
I was thinking about your individual differences study, and I thought that was a really nice thing to try. But actually, your data came out the way I might have thought it would come out if what you're looking at is individual differences in ability to gut sense. Mm-hmm. So rats which have good gut sensing might acquire a preference quicker or show up a stronger preference, but at the same time would sense the nutrient effects of overconsumption. So on the cafeteria diet might be more resistant to overeating. And I'd wondered if you'd thought about it that way.
Yeah, so that effect really needs to be explored and unpacked. I'm hitting all the academic buzzwords in my talk today. I talked about the extent to which and that things need to be unpacked. I have no reasonable grounds for speculation about the nature of that individual differences. It could be an individual difference, as you say, in gut sensing. It could also be an individual difference in associative learning. So it would be worth investigating, do individual differences in that flavor nutrient acceptance also correspond to other differences in associative learning outside the food domain? Mm-hmm. Because one of the reasons I got interested in that question was because of all of the work relating individual differences in cognition to obesogenic diets. So the sort of failure of appetite control that you get as a result of overeating an obesogenic diet being a byproduct of hippocampal learning deficits. And so, your suggestion is a really good one. And it's something I would really like to explore more. I think I saw Harvey Grill on the call a while ago. I don't know if he's still on, but Harvey did mention to me a while back when I told him about these data, some preliminary data from his lab on individual differences in rat satiation responses, I think to CCK, and how that predicted responses to an obesogenic diet. But I'm going off of a memory of a conversation at a conference or at a cocktail party, actually. So
that paper is published, actually.
Oh, it is?
Yeah, it's Hallie's. Hallie's paper.
Those are Hallie's data. Okay, yeah.
Yep.
Thank you.
Mm-hmm.
Kevin,
yeah.
I know we're way over time, but I have one last quick question. Mm-hmm. Has anyone looked at, is dopamine signaling required for this flavor nutrient learning?
Mm-hmm. Okay, so I will confine my answer to the rat work because I know that Dana Small and her group have been doing a lot related to this in humans, but I know the rat work a little better. And again, it's Tony's work with Rich Bodnar, that micro-infusions of different, or Tony, I'm sorry, I don't remember if these are systemic IP treatment or micro-infusions into different spots in the mesolimbic dopamine system that interfering, at least in rats, interfering with dopamine signals during acquisition of the learning. It prevents conditioning, but manipulating dopamine signal after the learning has occurred at the time of testing doesn't seem to interfere with the behavioral expression of preference. But you know, at that time, we weren't as tuned into sort of these subtler behavioral measures of, say, rapid feedback effects within a meal, and how rats might respond early on in training rather than with overtraining. So there's a little bit of work that, as a small amount of work, points to the solid conclusion that in rats, dopamine is crucial during acquisition, but less crucial during later expression of what's already been learned.
And does that extinguish?
Okay, so flavor nutrient conditioning does appear to be, okay, some people say resistant to extinction. And I have some unpublished data showing it's not really as resistant to extinction as we think it is. After repeated exposure to the flavor no longer paired with nutrition, rats will still continue to express a strong preference for it over a CS minus. But if you test it relative to another CS plus that has not been extinguished, the preference for the extinction trained one drops. So that is a little bit of a mystery. Why after extinction training, it remains preferred over an unpaired stimulus, despite the fact that it's lost value relative to what it used to be.
Great. Any other questions for Kevin?
Can I chime in? Tony Scafani.
Mm-hmm.
Kevin, you correctly pointed out that rats and mice differ substantially. So whereas rats are poor learners with fat, mice learn fat preference as rapidly as glucose. So they're
Mm-hmm.
interesting.
Mm-hmm.
Also, in response to Mark Friedman's question, MCT fat is less effective than LCT fat in rats, but we haven't done that in mice. And finally, your distinction between early and late signals is very interesting. And one neat way to investigate that, at least in the mouse, it won't work in the rat, is to compare the effects of intragastric infusions of glucose versus alpha-methylglucose. So alpha-methylglucose activates SGLT1,
mm-hmm.
but doesn't raise plasma glucose, so doesn't generate the post-absorptive signal. And so MDG, alpha-methylglucose, would likely only generate the rapid signal, not the delayed signal, and glucose would would generate both. So you could do your early and late meal studies in the mouse using those two kinds of glucoses, and I think that would be a very productive experiment.
That's a great thought, Tony, and that had actually occurred to me once before, looking at it in rats with the early and late, because do I recall correctly that in rats, alpha MDG produces a mild avoidance or a possible?
Yes.
That my thought was, if it hits that early, so if we did the early and late, it might effectively hit that positive early appetition signal, but then is also producing a more delayed aversive signal that's swamping learning about the earlier signal. And if we did it in the early late paradigm, it might reveal that. So I think it's worth doing in rats as well as mice. Thanks, Tony.
Kevin, thank you so much for a really fascinating seminar.